Hi there, I want to share with you my research area, which is protein refolding dynamics. Proteins are complex biomolecules that plays important roles in many biological processes. To function properly, proteins must adopt specific three-dimensional structures that are dictated by their amino acid sequence. However, proteins can become denatured or misfolded under certain conditions, such as exposure to high temperatures, pH extremes, or chemicals. Misfolded proteins can be toxic to cells and are also associated with many diseases, such as Alzheimer's, Parkinson's, and cystic fibrosis. The figure on the right shows the process of Alzheimer's disease due to misfolding of A-beta amyloid. Therefore, the studying of protein refolding mechanism is crucial. Then, lysozyme is a small enzyme that plays an important role in the immune system of many organisms, including humans. It was first discovered in 1922. Lysozyme is found in many bodily fluids, such as tears, saliva, and mucus, as well as in egg whites. Lysozyme is a relatively small protein consisting of 129 amino acids, and it has a compact globular structure. The enzyme has a deep cleft in its structure that binds to the trains of bacterial cell walls, and its catalytic site contains a cluster of amino acids that act as a proton donor and acceptor during the hydrolysis reaction. Lysozyme has many applications in biotechnology and medicine, including as a preservative in food and as a treatment for bacterial infections. It is also a model system for studying protein structure and function, as its small size and relatively simple structure made it a useful tool for understanding the fundamental principles of protein refolding and enzymatic catalysis. Next part is experimental section, but my work is mainly in the technique calculation part, so I will only briefly introduce the experimental process. There are two main processes: denaturation and renaturation of lysozyme, and the monitoring of this process. We use the enzymes activity assays and HPLC assays. And this is ChemE representation of the simplified in vitro refolding process. Proteins that are denatured and repeatedly converted to intermediates, which are subsequently depleted through competitive or pathway refolding and off-pathway aggregation reactions to produce native and aggregated proteins. When it comes to lysozyme refolding, the conversion from D to I um, occurs within a few milliseconds, allowing for a further simplification of the refolding techniques modeled into simultaneous formation of native lysozyme and aggregates from transient intermediates. Refolding of lysozyme is a first-order reaction since it revolves a single molecule. However, the formation of aggregates is of a higher order because it is caused by intermolecular interactions. Therefore, the reaction rates for native lysozyme formation and aggregation are given by the following equations. So the following differential equations were used to describe the refolding techniques. The model equations were solved numerically. A program was writing on the MATLAB Pro platform. The ordinary differential equation was numerically integrated with ODE 45. It's an ODE solver embedded in MATLAB. And then let's talk about conclusion and discussion. The graph on the right shows the fit under optimal protein refolding conditions, with the dots representing the experimental data obtained from monitor at different times. The lines present the fitted data. The SSE value of the fit was 0.237. The value of R squared was 0.987. Related papers show that in the lysozyme refolding reaction, the order of the aggregation reaction decreases as the reaction time progresses. In our simulations, the order of the fixed aggregation reaction is 3, which makes the model less accurate. The fit is not ideal. Therefore, we thought about how to further optimize the modeling process. Thank you for your listening. That's all my current research.